Now, up until this point in our cellular respiration lectures, I've been using glucose as our kind of standard biomolecule for energy production, right? It's all about glucose going through all of these different processes, glycolysis, then the Krebs cycle or fermentation, depending on whether or not there's oxygen present, followed by the citric acid cycle, followed by oxidative phosphorylation. However, glucose is not the only fuel source that is available to your body. And in fact, you virtually never eat just regular glucose molecules. I mean, even when you're eating sugar, table sugar is made out of sucrose, which is a disaccharide. It's one glucose plus one fructose held together by a glycosidic bond. So you actually get most of your calories through sucrose, other disaccharides, some polysaccharides like starch, or through fats, through proteins, all of the other elements of your diet. If I were to eat like a whole bowl of almonds, for instance, I would be getting protein. I would be getting oil, so fats. I would be getting sugars. I would get starch from that as well because a lot of starch is stored away inside these almonds. But I'd be getting very little raw glucose. So the reality is a wide range of different biomolecules can actually be fed into this same cellular respiration pathway that we've been using glucose to talk about. The easiest way to see this is to think about digesting starch or glycogen. I mean, those molecules are just made out of individual glucoses which have been linked together. So by hydrolyzing the bonds between the monomers, you can get glucose, and then you can follow cellular respiration exactly as we've learned it so far. But fats also serve as an energy storage macromolecule. If you remember the structure of a triglyceride, you have these fatty acids, which are hydrocarbon chains, so there's lots and lots and lots of hydrogens on them, and those hydrogens have high energy electrons, just like the glucose molecules we've been talking about, and they're attached to a glycerol molecule, which is kind of sugar-like by itself. So the first thing that your body's going to do, the enzymes in your body are going to do when you eat a lot of fat, is it's going to start hydrolyzing the bonds between the fatty acids and the glycerol molecule. That glycerol molecule can be converted directly over to G3P, which is an intermediate in the process of glycolysis. Meanwhile, the fatty acids are going to be broken up into 2 carbon units. They're just going to be snipped apart so that each little segment of it only has 2 carbons, and then that's going to get picked up by coenzyme A, and we're going to use those fatty acids to produce a whole bunch of acetyl-CoA, which is the chemical constituent that enters the Krebs cycle. Now, fortunately or unfortunately, depending how it is you want to look at it, fat contains about twice as much energy as an equivalent mass of carbohydrate. That is to say, you can generate about twice the amount of ATP by breaking down a gram of fat that you can by breaking down a gram of carbohydrates. What that effectively means is that your body doesn't actually have to burn all that much fat in terms of sheer grams or sheer pounds of mass in order to meet your energy needs. So if you've ever struggled with weight loss or known anyone who has, there is a very good chemical reason why it's so difficult to burn fat. Whereas if we stored a similar amount of energy on our bodies in the form of, say, glycogen, it would be a lot easier to lose that weight. Now, typically your body doesn't consider protein to be a rich energy source. It wouldn't be your body's first choice to get calories out of. If you're going to break down a polypeptide into individual amino acids, most of the time what you're going to do is take those amino acids, string them together in a new order inside your own cells, and use it to make some of your own proteins. 50% of the dry mass of any organism is going to be protein, so you need lots and lots of protein in order to build up your cells, build up your body, and conduct all of these metabolic processes that we've talked about. Remember, every single step required an enzyme, and just about all the enzymes are proteins. We are going to talk about a situation in DNA processing and getting the information out of DNA where we actually use RNA enzymes, but most enzymes are proteins. However, When push comes to shove, your body, your cells, can also take those amino acids, they rip off that amino group, that NH2 group, it's called deamination. They remove that, and then they can convert what's left of that amino acid, or what used to be an amino acid, into some of the other chemical constituents that have entered the cellular respiration process at various points. Amino acids can be converted over to pyruvate, and they can be converted over to acetyl-CoA, and they can be converted over to other intermediate products of the Krebs cycle as well. So the purpose of cellular respiration is not just to break down sugars, it's also to break down and get energy out of any kind of organic molecule that you're taking into your diet. Another important concept that's linked to this idea that you can feed in all of these different kinds of molecules to the cellular respiration metabolic pathway and get the same kind of results is that if you reverse that process, if you take it and flip it around backwards, that this is in fact reversible, that you can actually take some of the chemical constituents in cellular respiration and then you can convert them over. You can make other kinds of biomolecules out of them. You can make amino acids. You can make polysaccharides or monosaccharide, whatever kind of chemical constituent you need. The cellular respiration process is a kind of molecular interchange. It acts as a kind of recycling center, allowing you to bring in molecules of any kind from your diet or ones you're not using in other parts of your cells or your body, and switch around the groups, switch around the atoms, switch around the functional groups until you've gotten some new materials that you might need in order to build up other aspects of your body or your cells, make new proteins, for example. Now, you wouldn't want your cells to be making all kinds of biomolecules kind of willy-nilly at random, or you wouldn't want them to be tearing down molecules that maybe you need or that maybe you don't need energy right now. Maybe you're all set, you don't need it to be breaking anything else down. So what your enzymes usually do to control this kind of metabolic process is called feedback inhibition. Feedback inhibition is a mechanism by which the products of one chemical reaction, of one metabolic process, actually inhibit that same metabolic process. Now, that's a little bit complicated. It's a bit of a mouthful. So let me see if I can give you an example. Let's say a cell already has entirely too much ATP. It doesn't need any more energy to be processed. It's got enough ATP for right now. ATP is kind of unstable, so if you can keep as much energy as possible in the form of glucose, that's better for long-term storage of that energy. You only want to make the amount of ATP that you're going to need right away. Well, if there's a lot of ATP kind of floating around inside the cell, it's going to be bumping into other kinds of molecules. And one of the molecules it's going to bump into is an enzyme that functions in one of the very early steps of glycolysis. And when ATP bumps into that enzyme, it actually switches it off. So as we get more and more and more ATP feeds back, it turns off the process that makes the ATP. So it prevents an excess amount from building up. Now, as we use the energy in the cell, that amount of ATP is going to shrink, and it's going to be converted over to ADP, adenosine diphosphate. When adenosine diphosphate interacts with that same enzyme, it actually switches it on. It switches it into hyperdrive. It says, oh, wow, we have a lot of ADP, adenosine diphosphate, around. What we actually need is more energy, because you only have so much of this coenzyme around. Some proportion of it is going to be in the form of ATP, and some proportion of it is going to be in the form of ADP. If you have way too much ADP around, that means you're lacking ATP, so it's going to switch that enzyme on. This is called feedback inhibition, once again, where the products of these chemical reactions, the products of the metabolic pathway, feed back, they go back, and they interact with some of the earlier enzymes to change how they are functioning in order to regulate. Remember, one of the criteria of life is regulation. This is a method by which organisms and cells can regulate their own internal metabolism. So the enzyme that we're referring to in that example is called phosphofructokinase. It's active in one of the early steps of glycolysis. If you have a lot of ADP or AMP, adenosine monophosphate, which is an even lower energy form of this coenzyme around, it's going to stimulate phosphofructokinase, and it's going to make it proceed forward with glycolysis, ramp up production of ATP. However, if you have a lot of ATP or a lot of citrate, which is one of those products of the Krebs cycle, then it's going to turn it off because we already have plenty of the chemical constituents around, plenty of ATP and plenty of material for making ATP already in play at that point. Another example of allosteric control is in fat anabolism. So anabolism is building up large molecules from smaller starting molecules. In this case, the large finished molecule that we're going to be producing are triglycerides. We're going to be making fat for our cells. Now, acetyl-CoA carboxylase is an enzyme which is important for making fatty acids, and you need fatty acids to make triglycerides, 3 of them for each triglyceride. However, the activity of acetyl-CoA carboxylase is affected by the levels of all kinds of different chemical constituents and hormones and other factors inside of the cell. The same chemicals that were affecting phosphofructokinase, citrate, and AMP also exert control over this particular enzyme as well. In this case, citrate activates the enzyme and AMP inactivates the enzyme. Now that is the opposite of what they did to phosphofructokinase. And that kind of makes sense because while you're trying to produce more ATP, it doesn't make sense to simultaneously be trying to build up more and more fat molecules. Fat is a long-term energy storage molecule, so if you have a lot of AMP lying around, you're depleted of energy, there's no reason to be storing that energy away, you gotta be using it. So the same molecules that turn off one enzyme could be used to turn on another one or vice versa. If you have a lot of citrate lying around, that is going to ramp up production of fatty acids, and it's going to decrease the amount of glycolysis because you clearly already have been doing a bunch of glycolysis at that point. In other words, if you have a high amount of citrate, it's a good indicator that the citric acid cycle doesn't presently need more fuel to push itself forward. Now, metabolism is a very complicated subject, and there's a lot of metabolic pathways we could talk about, and there's so much more content that we could discuss, but this is not a biochemistry course, so it has to come to a conclusion sometime. So let's review the differences, the similarities and the differences between the 2 major metabolic processes that we discussed, photosynthesis and cellular respiration, before we move on to any other kinds of content. Photosynthesis is an anabolic process. We build up materials, taking carbon dioxide and water and making glucose. Cellular respiration is a catabolic process. We're breaking materials down. We're taking glucose, and now you've seen other types of organic molecules as well, and we are breaking them down and we're releasing CO2, we're releasing water, but we're also releasing energy, which we can store into the form of ATP. In terms of oxidation reduction reactions, in photosynthesis, carbon is reduced. We go from CO2 to C6H12O6. Carbon has a lot more hydrogens attached to it than it did before. And in biological reactions, when you add hydrogens to something, that's usually reduction. Whereas oxygen is oxidized, and that is staggering. That almost never happens anywhere else in biology. Oxygen is a very powerful oxidizing agent. It is not itself oxidized under most circumstances. That's how powerful photosynthesis is. In cellular respiration, it's just the opposite. Carbon is being oxidized and oxygen is being reduced to produce water. The location of photosynthesis is in the chloroplasts, at least for eukaryotic organisms like plants or some kinds of protists. The light-dependent reactions occur on the thylakoid membrane. The light-independent reactions occur in the stroma of those chloroplasts. With cellular respiration, the location is the mitochondria. The process of glycolysis happens outside of the mitochondria, but pyruvate oxidation, the transition reaction, the Krebs cycle happens in the mitochondria. The Krebs cycle happens inside of the mitochondrial matrix, and oxidative phosphorylation also happens in the mitochondria on the inner membrane. Across the mitochondrial inner membrane is where oxidative phosphorylation happens. That's the electron transport chain. Both of these things have an electron transport chain. Photosynthesis is we have the light reactions. The light-dependent reactions involves an electron transport chain. That's where we had the photosystems and cytochrome B6F and NADPH reductase. And in cellular respiration, we have oxidative phosphorylation, which we just finished going through. There's reactions happening in the cytosol, and both of them are a chemical, a metabolic cycle. The Calvin cycle happens in photosynthesis, and the Krebs cycle happens in cellular respiration. The overall chemical reaction is a mirror image of each other. You just got to flip the arrow around. 6CO2 plus 6H2O plus energy in the form of light gives you C6H12O6 plus 6O2, gives you oxygen and glucose, whereas with cellular respiration, C6H12O6 plus 6O2, glucose and oxygen, can be reacted together to give you 6CO2, 6H2O, and up to 38 ATP, as we just saw, if you combine all of those different metabolic processes together for cellular respiration. And these two processes in eukaryotes are both managed by bacterial endosymbionts, organelles that started their evolutionary history as their own free-living bacteria, and yet even though they were incorporated into eukaryotic cells at entirely different times in evolutionary history, they managed to end up with mirror image opposite kinds of energy processing jobs, which is kind of remarkable.